Our next speaker is Ton Logtenberg. We know Ton for his work in Merus and Visa, companies that he founded over 20 years ago. And he served as a CEO, president, and principal financial officer since he took this company public in 2006. This company is working on exciting bispecific molecules. I believe there's a lead product and currently in phase 2 and NRG1 fused cancers. And today, Ton will tell us about re-evolution of antibody therapeutics. It sounded a little bit like Matrix Reloaded, and I'm very much looking forward to his talk.
Thank you very much. And thank you for inviting me to this great meeting for celebration. I intend to speak a few words of congratulations at the end of the talk. But before jumping into my own presentation, I want to make two short comments. And the first one is really about the title of this conference, which has the word series in it, which of course raises the expectation that we will see each other again here in this beautiful town again. And in the pure hypothetical situation that I'll be invited to the organizing committee, I would suggest that we will organize a tour through the Douro Valley so everybody can taste the wonderful Portuguese wines that are very nearby. And the second short comment is about the recent joining of efforts of Iontas and FairJourney. And I think we are looking at an even more powerful antibody company that can do many good things in a world where antibodies become more and more popular. And as Greg mentioned, the good news for FairJourney is that they get 50% of John McCafferty. Now, when you look at it, 50% is enough for conveying lots of wisdom to other people, right? You only need maybe a Friday afternoon drink. That's often enough. But the even better news is that John's 50% will also bring in his incredible background in antibody engineering. He's been of the true pioneers in the beginning. And it will also bring his Scottish humor, which is quite interesting, as well as his undeniable talent to play the bagpipes. And of course, us standing on the outside will look at this experiment going forward and are very eager to see what the outcome is going to be, the British version and the Portuguese version. Where will that get us? Okay, so back to antibodies. I'll give you my view of the antibody world. And maybe not surprisingly, somewhere halfway, the organoids will come in. So while you are recovering from Hans' story, I can tell you we recovered years and years ago, being very close to the organoids. And of course, we incorporated them into our systems. And just to give you one of the take-home lessons is that they're not only good for the diagnostics and the wonderful things that Hans has described, but also very attractive for actual drug discovery at a very early stage. Okay, I'll get back to that a little later, but organoids will be back. Okay, so this is my outline, very simple. So I'll talk a little bit at where we come from in one slide. Greg has already given a good overview of some historical hallmarks, but I'll give you my own version. And of course, where we are today, and then where we are going. So this is a small table that I think, from my perspective, really are hallmarks and that have driven this field forward and have come to where we are today. And in essence, of course, it's the hybridoma technology that started in 1975 with the Köhler and Milstein paper, and then, of course, the first murine antibody that was injected into humans in 1986. I think there was a point in time where we started to realize that although these fantastic monoclonal antibodies were very attractive because of all the goodies they bring, they're also a challenge because they're not human. And I think that was really the start of an enormous effort from antibody engineers and protein engineers in general to try to come up with solutions that would make, at the end of the day, a human monoclonal antibodies. And of course, you see a list here where we've seen phage display, we've seen yeast display coming up, We've seen the Fc engineering to make even better antibodies from their effector function point of view, and a whole series of antibody fragments that long part as part of the phage display efforts, but also as drugs that could have unusual properties when applied in humans. So that is, I think, in a period of 20 years, there's been done an enormous amount of work to get to these true drugs that are attractive from a manufacturing point of view and behave well in humans. Now, we, at least being in that field for a long time, I would call that a revolution, right? What's 20 years? But of course, we all realize that investors and analysts will call this an evolution. It takes forever. These guys don't have their acts together. It takes them 20 years to get anywhere with something that looks like a drug. And that's, I think, where sometimes the tension is between investors putting a lot of money, calling something an evolution takes forever, where we say, well, we're making great progress, and these are challenging problems we're working with. And then, of course, my particular view on where we are, what we have done in 2020, in the period of 2000 to 2021, is really the multi-specific formats. The rest of my talk will focus on multi-specific formats as next-generation antibody drugs. And I think it's good to mention that we have now more than 100 unique therapeutic antibodies that are approved as drugs. So this is, and also highlighted by Greg, is a very important drug category. Now, what other outside things out of the antibody space have actually contributed to where we are today? And I call this increasing complexity that is driven by a number of different facts. First of all, the antibody formats. There are so many to choose from these days that it becomes a very big challenge to make a decision. Mechanisms of actions have increased over time. So for now, to consider a product, you have to really think about more mechanisms of actions. The number of cell types involved has increased dramatically, right? In the old days, you would think about a cancer cell, that's your target, a couple of receptors that control the growth, and you were done. Now it's cancer cells, it's T cells, NK cells, endothelial cells, macrophages, fibroblasts. There's a whole universe of biology that's coming at us in which we try to understand whether these processes are druggable and actually make a difference for patients at the end of the day. So the choice of cell types and molecules involved has dramatically increased, and has life not made necessarily a lot easier, for that matter. And I remember complaining in the old days that everybody was talking about TNF and CD19, and that was a bit boring at some point. And now we have so many targets and target combinations to think of that we have yet quite another problem. So the last to mention in this list is the incredible technical revolution we've seen, especially in DNA sequencing and RNA-seq approaches, where now it has become possible to take single cells that are relevant for disease and single cells from multiple patients to actually look at targets, to look at overexpression, and to come up with a whole new host of potentially important targets that drive biology or pathology. So all of you working in this space probably recognize this list and keeps you awake and maybe makes you think back of the CD19 TNF days and think about simple things to work on. It's not simple. The formats are not simple. The drugs are not simple. And I think one of the other take-home lessons is that we do need much better preclinical models to work on to get to mine through all these possibilities. So I'm now going to my pet subject, which is multi-specific formats, because I think based on what I just said about the complexity, it will also drive the need to develop drugs that actually have multiple functions that are assembled in one drug, right? We cannot inject 7 or 8 drugs into a patient to conquer various parts of for example, a cancerous process, but rather we need to have drugs that do multiple functions all in one, so we can keep combining and just add to the list of activities that we want to take place treating a patient. And I think multi-specific formats may be a solution for the future for that. Okay, so Just one little bit of history here. It's hard to realize, but the original concept of bispecific antibodies was already defined by Nisonov in the 60s, right? This is way before monoclonal antibodies, but the concept of two different binding sites in one immunoglobulin molecule is actually quite old. And since, and this, I think the counting stopped last year, but we have seen more than 3,000 research publications, 100+ clinical trials that are currently running, and we have only 3 marketed products so far. Is this a good sign or a bad sign? I think looking at beginning of 2000 and 20 years of multi-specific format research, it pretty much copies what's happened to monoclonal antibodies, right? From the early realization of a monoclonal antibody can be a drug to that picking up, we're at the end of the 90s, early 2000s, where actually monoclonal antibodies start to hit the market. So 20 years for early technological breakthroughs and discoveries to drugs that make it to the market is not an unusually long time. So next time you talk to investors and they say, where are the products? They say, we're in good shape. We're doing well. And I'm sure looking at all the clinical trials that multiple multi-specific drugs will come to market in the next few years. All right. You probably have seen some version of this slide taken from the Contement paper, which defines the zoo of bispecific antibody formats. And it actually is a zoo. Hundreds of different formats have been described, are being used in many ways to develop drugs. And again, we all have kids that have a problem that's called stress of making choices, too many choices, and that's stressful. And I'm sure this is stressful for everybody that wants to engage in a particular format. It's very difficult to say, this is the one I want to work with, because at the end of the day, I don't want a construct, I don't want an approach that causes a late crash in my product, either in the clinic or in CMC, right? It needs to be robust, it needs to be proven, and then you can go after the second part of these difficult stories, which is the biology. Don't worry about the construct. Don't worry about your multi-specific antibody. So why were there so many bispecific constructs, or are there so many bispecific constructs? Because antibody engineers just came up with many different solutions for a particular problem that needed to be solved. So what that problem is, I'll just highlight in the next slides, but it is many different solutions to a particular problem. Another reason is often, just try to circumvent patents. When the full-length IgG molecules are patented, you make a fragment and call it something else, and then you may be circumventing a patent. And the other one is, of course, is to really try to incorporate desirable properties in your molecule that may even be better than what an antibody, a full-length IgG molecule, can bring. So in this whole endeavor, we've really moved in the last 20 years from a technology-driven phase where the hurdles to come to products that behave well in patients were solved, to an exploring these new modes of action, for example, T-cell engagers, to now in a very complex area of biology where target selections in conjunction with selecting the right format, biggest challenge going forward. Just a problem to solve very, very quickly. Most of you know about this. But in the old days, 2 hybridomas were fused to make a quadrobid or hybrid hybridoma. And what happened is that in addition to the desirable bispecific, which is shown here in the 2 different colors with the correct pairing of the heavy and light chains for the different specificity in one molecule, you would be stuck with all these contaminants where something was wrong with each of these molecules. Often a mispairing of the light chain, homodimerization of the heavy chains versus heterodimerization, and of course that would create a mixture of antibodies that would be inseparable and would not ever generate a way forward to creating products. So one solution that Mirus, the company that I founded, pursued was actually based on a common light chain. And what does that look like? A common light chain means that every antibody in the systems we use always has exactly the same light chain. There's just one light chain in the system. And that, when again we go to the desired antibody, where the two different heavy chains and the common light chain incorporated in the single bispecifics, you're only stuck with two contaminants, the parental molecules. So now we're moving to a less complex situation where the common light chain takes care of many of the issues with the hybrid-hybridoma approach, and just we need to get rid of these parental monoclonal antibodies. And that was taken, that solution was provided first by Genentech. Genentech came with their Dob and Halls approach, in which they actually force paired the two different heavy chains by what they call the Knob and Hall technology, where one heavy chain would have protruding amino acids, and the other heavy chain would have amino acids with fewer, if any, side chains that would actually accept the knob as a whole. And that was the first attempt to drive heterohybridization. At Mirus, we used a different approach. We looked at the same region, the C3 region of the molecule, and we came up with changing just a few amino acids, 2 on each heavy chain, to drive heterodimerization by electrostatic attraction or repulsion. So plus attracts minus, this will repulse minus, and plus will repulse minus. And at the end of the day, after doing many experiments in the group, we came up with a combination that gives a virtually 100% bispecific just after transfecting 2 different DNA constructs into a single cell. And that is what we call now the biclonics molecule. It has these 2 changes in the C3, use the electrostatic attraction, and we work with a common light chain approach to get rid of the mispairing of the VH and the VL regions. And then this platform was further extended to do everything that we want to do with a monoclonal antibody, is have ADCC enhancement or a completely silenced FC region for particular applications. Now in the early days, so just to focus on one aspect of this platform a little more, is a transgenic mouse that we created a long time ago. This mouse was different from the conventional transgenic mice that had been generated by Medarex, for example, or by Regeneron in their first versions. This mouse was engineered to make these common light chain human antibodies. And this is a short cartoon on how that was achieved. So first of all, we knocked out the endogenous heavy and light chain loci in these mice, and then introduced these rearranging heavy chain mini loci that have the VDs and Js that you find in the human genome. We kept the constant regions of the original species to get correct B cell signaling and to get correct processing during B cell development. And of course, the common light chain was reintroduced as the only light chain in the whole system. And this was done in a fashion that this light chain was resistant to somatic hypermutation. So essentially, it stays in germline. We know exactly what the light chain is. We did multiple MIMO lines to incorporate some variations on this theme, but at the end of the day, this mouse now has generated more than 40 antibody panels. We know, despite the fact that we only have one light chain, this mouse generates very high quality antibodies that have all the affinity ranges, the epitope diversity, the somatic hypermutation, everything you want from a transgenic mouse without losing anything by just introducing one light chain. And that was, of course, a question in the early days. Immunologists like to think that you need all the diversity at the light chain locus as well to get the best possible antibodies. Okay, now another principle of the company is that this mechanism of action that we're after, how do we best get to the best leads? And the basic choice to make, do we work with a few antibody candidates that we have sitting around in our refrigerators and are easily accessible to start with, or do we go a completely different approach which says we are actually testing thousands of bispecific antibody candidates in functional screens and not be too biased about that we understand enough about the targets, the epitopes, the affinities of the arms, and all of that. And we actually, over time, became very modest about understanding how antibody works. We can talk about this forever, but functional screening sort of solves that problem. You let the biological assay tell you which ones are the ones that work, and then try to understand later why they worked. So I'm going to highlight this principle in 3 different programs that are, 2 of them are in the clinic, and the 3rd one is moving along. And just to show you what exactly we did to get to these leads and make these thousands of bispecifics and mine through them. And this is in essence the functional screening approach. So you do make these enormous panels. These panels, before they are assembled into a bispecific, have been characterized to a certain extent, for example, by binning, such that the arms all bind to different regions of the molecule, nothing overlapping, different epitopes, different affinities. They're combined in transient transfection into a 96-well plate or 24-hour well plate to get the production. And here is where the efficiency of the heterodimerization comes in, right? You need about 100% bispecific molecule to come out of your cell to just take the supernatant and to run your functional assay. If that's not right, you have a mixture, you're going to get mixed results in the assay. Functional screening, typical readouts that we get, a lot of data points that make us go from thousands to 50 to 10 to 1, and eventually take that lead forward into the clinic. Okay, so this is the first one I'm going to describe. It's, the lab name is MCLA-145. The paper was just published in Nature Communications a couple of weeks ago, and I'll just take you through the process of thinking and selection to give you an idea of what we want to do. And this is a bispecific that binds to CD137, already introduced by Greg on one side, and binds to PD-L1 on the other side. So the original thought was, let's use functional screening and bispecific antibody panels to make combinations that will address some of the hot targets in immuno-oncology that we would like to combine into one molecule and to see whether we could get additional biological activity from these bispecifics. And in these, of course, we all know about PD-1, PD-L1. CD137 is indeed an inducible co-stimulatory receptor expressed on activated T cells and NK cells. So for this talk, I'm focusing on the combination PD-L1, CD137, and just to give you some more background information on what has been done so far with this target, there have been 2 monoclonal antibodies in the clinic that are pointing for various reasons. One of them gave liver toxicity, unacceptable liver toxicity, so that was really a showstopper for that molecule. And the other molecule was actually displayed disappointing agonist activity and also didn't really make it going forward. But there was a mechanistic rationale for combining these activities from preclinical models from mice. In essence, by combinations of monoclonal antibodies, it made sense to go after bispecifics and see if we could solve these issues. So here is the basis for these experiments. For this particular approach, we made 450 different biclonics, combining the CD137 arms, either with PD-1 and PD-L1. And in the first screenings, we actually saw very disappointing results because we didn't see any activity of these molecules, actually expecting that we would see something based on the combination CD137, PD-1, because these are co-expressed on T cells. But it didn't pan out that way. In a reporter assay where the URCAT cells actually express both targets. And then we moved on to add to these assays a CHO cell line that was transfected with CD137. So we have a second cell type now, and we're looking for trans activation conveyed by the molecule, whether the first attempt was cis activation on the same cell. And here's just one of the readouts. You can see on the left the CHO cell that has been transfected with PD-L1, and on the right control, the CHO wild type, and the urilimab as a positive control with very potent agonist activity is in the graph as well. And you can see some of these top lines and also lower lines, but they in general depict the kind of variability that you're getting. You see in the top lines very potent activation of the reporter construct based on the presence of PD-L1 on CHO. Now, that was all by itself already a bit exceptional, because, and I think Greg mentioned that already, the ligand for CD137 is a trimer, and you need the trimer, and you need the typical trimer-driven activation of CD137 to get activity. And these are two monovalent arms, right? So you wouldn't really expect the monovalent arms to drive this. But after a lot of experiments later, we actually realized that binding to PD-L1 would cause microclustering on the PD-L1 positive cell that would also then cause microclustering of CD137 and cause activation. So it's a new mechanism of action that was a bit against the dogma that you always need these trimers. So we did a lot more work, so I'm just summing up a couple of data here. But first of all, we identified a completely different epitope that was potent in any bispecific that were carrying this arm. It was completely different from the epitope of the two anti-137 antibodies that were in the clinic. And it was also an epitope that blocked the binding of the CD137 ligand. Directly from the mouse, so the monovalent affinities were high, 0.5 nanomolar and 1.9 nanomolar for the PD-L1 and the CD137 arm. And the mechanism of action, as we now see it, is actually context-dependent activation of T cells, which means you need PD-L1 on a tumor cell before the activation takes place. The PD-1 arm all by itself blocks PD-1 interaction, so that's another functionality. And the third functionality is that by mechanisms we don't fully understand, the bispecific antibody attracts new T cells to come into the tumor. So I think here we created a molecule that has different attractive functionalities in one cell. This molecule is in the clinic. It's gone through dose escalation. We know it's safe and well tolerated. And in the near future, we'll hear about the phase 1b extension data that are now being generated at Mirrors. Just one more piece of data before I close this subject. But again, this molecule is potent in many different assays and not only activates naive CD8 positive T cells, but also CD4 positive memory T cells. And I urge you to go to the original paper from the Mirrors group to look at all the details of what exactly are we doing here biologically. So CD137 PD-L1 bioclonics is an example of how functional screening, large numbers of bispecifics, get you to a molecule with unusual characteristics that indeed we would probably not have designed if we were just taking our knowledge at that point in time and try to create molecules by stitching arms together rather than following the functional screen. All right, the next program is called A158, also in the clinic. This is where the organoids come in, so you won't be surprised that one of the arms is binding to LGR5 and the other arm is to EGFR. So Hans already extensively explained the virtues of the organoid system. We engaged early on and asked the question, can we actually use the organoids as better models for real drug discovery, antibody discovery, combining our functional screens with the organoid technology. And this work was a collaboration with Hans's lab, with Eduardo Battle, who was in Barcelona, a former postdoc of Hans, and Mirrors. And there was some evidence that LGR5 was actually an attractive target from the Genentech group, for example, who showed that LGR5 is critical for liver metastases and also for regenerating primary tumors in animal models. But we took the sort of agnostic approach. We didn't really zoom into LGR5 to begin with, but we made panels of common light chain antibodies through MIMO against a number of Wnt targets that are shown here, as well as a number of receptor tyrosine kinases, and made all the combinations again to see whether we could come up with molecules with desirable properties. So some comments here on the organoids. I think we also took the stance that indeed organoids are better models, better representative of what happens in these patients. Of course, being close to the Ubrecht Institute and the Hub Foundation that we founded, we also had access to these large biobanks of well-characterized colorectal organoids that would help us not only do the initial primary screen, but also do the validation going forward. And this is just an example of how this organoid biobank is characterized. In the top horizontal row, you see different organoids. And on the vertical axis, you see mutations in genes that have been identified in these organoids. And these are long lists where you can mine to see whether you can select particular organoids that give you data that make you think that you know what you're doing in the context of real tumors. Okay, another feature here is what we introduced is high-content image-based screening. So rather than looking at simple assays, we used this particular approach to assemble for each well 300 different data points that collectively gave us a better idea of what was going on in these cultures when they were treated with these bispecific antibodies that we were introducing. And this is just a very sophisticated way of not just looking at are we inducing apoptosis or are we killing cells in another way, but rather look at, for example, morphology of lumens, looking at complexity of lumens that can be large, that can be small, that can fuse, that can anything that you can measure through this high-content image-based screening helps you to identify the leads. And on the right, you can actually see that these organoids, depending on the growth factors that you add, may already have different morphologies and characteristics to begin with. So in the top wing, you can see the organoids grown in EGFR. On the bottom right, you can see them grown in neuregulin or in EGF and neuregulin on the top. And then the flow of cells that start to change is going into the lower left part of the diagram. And the number of data points that we assembled on these kinds of cultures is enormous, but it gives you a sound basis to make your choices for the leads that you want to progress with. So this is a slide that I took from my deck from talking to investors, just to impress the hell out of investors, saying, listen, this is how carefully we look at our data to define which of these molecules are the ones that we want to take forward. And these are incredibly large numbers, but all manageable in a system where you have the right components present in your lab to actually do this. So this is one of the graphs that I used to show why we picked MCLA-158. So this is in a validation screen, and 158 came out as the best molecule, giving 60% of the organoids. Here we saw the first glimpse also that this bispecific antibody worked for those organoids that have KRAS mutations, which are usually particularly difficult to address. And this is one of the final slides for this program. So on the left, you can see a data point that tells you that this is a molecule that works in situations where cetuximab has no activity. So the bottom line is MCLA-158 in a killing curve, and cetuximab here doesn't show any activity compared to the normal control. And on the right, you see another representation of what got us excited and drove our discussion, our decision on taking this molecule into the clinic. In the top 5 rows, you see normal organoids from the same patients from which we later derived the organoids from the tumor. So this is from adjacent tissue that's called normal by the pathologist. And you actually see in these normal organoids, this compound 158 hardly shows any effect, like cetuximab, right? The green and the yellow there show that there is weak or no inhibition at all. But if we then take the tumor organoids, and you see the red and the yellow on the right, these are where the inhibition is very potent, and again, it has little activity. So this had the promise that we were not addressing normal tissue with this compound, so no problem for normal LGR5 positive stem cells, apparently based on the organoid data and very strong activity in the tumors. So again, I want to stress, and Hans already told this, but I think organoids are not only good models to do your drug selection, but also drug validation, and perhaps to highlight again, as a method you can use in conjunction with running a clinical trial, because if you have outcomes out of your clinical trial and you have the associated organoid in your lab, you can, for example, already start working on combination treatments while you're looking at the outcome of the patient trial, right? It is a way to actually create the model in your lab while the patient is responding to the drug or not, and get an idea how you want to pursue your clinical trials. I think that's another great application of the organoid model. Okay, I now move away from biclonics, and actually 158 is also in the clinic, and again, the company has just announced that they will give an update on the extended phase 1b data later this year. So interesting times. I now move on to an extension of the biclonics platform, which we call triclonics. And this platform was originally developed for T-cell engagers for solid tumors. But when you use your imagination, you can easily see how this platform can also be applied for addressing 3 different targets in all sorts of ways and variations. So just to highlight what the molecule looks like, it's the old-fashioned bispecific antibody biclonics that I just described. And we simply extended one of the arms with an additional Fab domain, again with a common light chain. So naturally pairing within the cell, driven by the common light chain. And we build in, this is John de Cruijff's group at Merus, we build in variable linker length to get some flexibility in that molecule. Because if you use a very short linker, you create something like a BiTE with little flexibility, and a longer linker where the geometry of the molecule can be completely different. Okay, this is the particular program here where we are working on, which wants to actually extend the potency of the T-cell engagers that are currently either a product like Blincyto, but also the many endeavors that are out there to use this approach for hematological tumors, which are relatively simple because the targets for the hematological tumors are well known and often don't cause any harm to the organism. But for solid tumors, we have few, if any, tumor-specific markers, and we have to come up with other smart ideas to solve that problem. So here we are targeting 2 different tumor-associated antigens on the tumor, where the CD3 arm, of course, binds to T cells. We have many different CD3 arms. There's a whole story to tell about how the CD3 arm affects not only activity, but also the cytokine release. So we've seen CD3 arms in bispecifics that actually cause massive cytokine release, and we have other CD3 arms that kill as efficiently but have hardly any cytokine release. I think that's a very interesting finding when you do the mining experiment and find out that not every CD3 arm is the same, and perhaps some of this cytokine release syndrome issues that we're seeing in the clinic can be solved by just using different CD3 arms. But I'm not going to talk about that for today. The combination is key here on the tumor cell, and the selectivity is based on this kind of thinking, whereby the individual arms binding to a target cell actually don't cause activation of T cells, but you need the binding of both targets, both arms getting engaged to get T cell activity. And I think it's known in the art that one of the solutions could be that the avidity effect actually carries you over the threshold of activation, whereas the low affinity monovalent interactions don't cause any activity. And this is how this works in real life. So you do your screenings. You see here 2 arms that have too high an affinity for the target, and you cannot discriminate between cells that only have a single target expressed or the target is expressed both targets are expressed on the cell. And here's an example of an outcome for in your screen where the activity of the single arm is very modest. These are the bottom two lines. And only when the two targets are present, you get a shift to the left, which means that at lower concentrations, you do actually get activity and engagement of and activation of T cells. So this is the concept. Transbinding, two targets, and a balanced affinity between the two different arms that drives the activity, as well as select the right CD3 arm, because not, as I said, these CD3 arms all give different activities. We need to test many to see which one works best in the context of this particular approach. And then antibody geometry, the Lincoln length may still also play a role. So again, one of the take-home lessons, screening is essential to interrogate antibody characteristics. And this is really what the job at hand. So we have these robotized systems, and we look for these hits in yellow in libraries that contain sufficient diversity in terms of the arms, the affinities, and the epitopes to find these kinds of hits. And this is what a hit looks like, and I'll enlarge it for you. But these are some of the antibodies and arms that we're using in these assays give this particular outcome, whereby only the combination gives highly potent activation of T cells at a low dose, and whereas the arrow denotes where the activity of the monovalent arm starts to kick in. And of course, the million-dollar question is this in vivo enough to create a therapeutic window? That's, of course, where it really starts to count. But I think this is a great starting point. And this is just enlarging part of that graph, showing that indeed we do have molecules that create this kind of difference. And when we take the two binding arms and we take another CD3 arm, for example, you can see again that that causes variation. You really also have to look at CD3 to make sure you get your maximum diversity, your maximum therapeutic window, if you wish. Okay, and this is one of our champion triclonics here, where you do see a 5,000-fold difference in activity based on the avidity effect that only kicks in when the tumor has both targets. Okay, so these are examples of how Merus addresses bispecifics and now trispecifics. And I think with a platform like this and your own creativity, you can come up with many more applications. Just make sure you have the right assay, and you do have the diversity in your arms to begin with, and robotize the systems to get to the right answer. Now, I wanted to finish up with some novel mechanisms of action that I think are worth exploring, because we try to see what the future will bring. We don't want to just keep doing the same thing. We don't want to end up in a situation that we had with TNF, with 7 or 8 products ending in the clinic. We're now seeing the BCMA T-cell engagers. I think 5 companies have phase 3 data. Everybody's immediately zooming in on exactly the same thing at a moment in time where the original sort of proof of concept is there, like in this case with Blincyto. So we try to keep advancing the field, and of course other peoples are doing the same thing. So I want to first of all here mention the biparatopic antibodies. So biparatopic antibodies are antibodies that bind to 2 different non-overlapping epitopes on the same target. The insight now is that actually you can be quite loose about the affinities of the individual arms. So don't make too strong a cutoff on that it needs to be sub-nanomolar or even low nanomolar affinity. As long as you have the right assays, you will find the right ones, and sometimes quite surprisingly. So I wanted to highlight a paper that came out earlier this year. It addresses the old problem of trying to inhibit CD73, which is a transmembrane receptor with enzymatic activity, and it had been notoriously difficult to actually get 100% blocking of this target. And this was a paper that actually selected only 11 antibodies, but they were all binned, so they had non-overlapping epitopes. There was some work done to characterize them. And if you look at their activity, and the colors give you the percentage of activity of blocking the enzymatic activity, you can see that the antibodies combined with themselves in a bispecific, so it's the control, actually show disappointing results. The antibody is not really capable of blocking the enzymatic activity in these assays. And when you do the screen, so you start to combine each antibody in the panel with each other antibody in the panel, you do find those as well, right? These are all bispecifics now, biparatopics if you wish, that have similar disappointing activities. But of course, the good news is that you see many that have very interesting activities. So this goes up to the categories 70 to 89%, as well as to the 90 to 100% categories in the dark green. And to my own surprise, in this particular approach, 25 actually showed quite attractive activities, and 29 were in the category of the similar activity of the parental monoclonal antibodies. This is good news, right? This is not a huge effort. This is a well-carried-out experiment and shows the potency of the biparatopic antibody to be able to completely block this enzymatic activity. So whether this is going to pan out when we're gearing up towards other biparatopic approaches, we'll have to see. The mechanism of action may be different. Sometimes the molecule gets locked into a conformation. Sometimes it blocks hetero or homodimerization. So my prediction is that a next wave of biparatopic antibodies with unusual properties will come along. And I think the first ones we're already seeing, Regeneron with a c-Met biparatopic ADC now in the clinic. The Zymeworks ADC antibody is also a biparatopic for HER2. And in those particular cases, the biparatopic actually causes very rapid internalization and into the lysosomal compartment for degradation. So trafficking, internalization are keywords associated with biparatopic antibodies. Another one is an approach that was also recently published by others, and it's called receptor inhibition by phosphatase recruitment, or RIPPER. And the concept here and the thinking is that even in the absence of ligand, many of these inhibitory receptors that carry the well-known motifs have a are phosphorylated to a certain extent. And that causes tonic signaling. And tonic signaling means that they give an inhibitory signal, for example, PD-1 expressed on a T cell. So the original thought was can we actually, and of course when ligand binds, you get the full kinase activity and inhibitory activity. So what happens with a blocking antibody, the ones that we have seen so many of, you can actually see that it will bring the receptor back to the tonic signaling state. Residual kinase, kinase activity, residual phosphorylation that causes suppression of the cell. Now, the bispecific approach I think is really elegant here, in which one of the arms of the bispecific binds to a transmembrane phosphatase, in this case CD45, and CD45 will be brought into the proximity of the target molecule, in this case PD-1, and then the residual phosphorylation of the inhibitory receptor will be completely removed. And this is a beautiful paper, lots of details about indeed that this translates into more potent T-cell activation, into very much delayed exhaustion, for example, and many other aspects that we know from regular blocking antibodies to occur. So keep an eye out for these kinds of approaches where a bispecific may be able to recruit other activities in proximity of your target. So this is a proximity-based assay, a proximity-based approach, and actually convey intracellular activities that may be very attractive. That's the broad concept. Okay, so that's my formal presentation. I think I made the point that things don't necessarily become easier with new cell types and molecules entering our space, and the need for the best possible preclinical assays to mine through many different molecules to get to leads that feel less like a gamble when you put them into the clinic. Okay, so I'm moving back to the reason why we are here in the first place. And we pulled from our files a picture from Fair Journey, of the Fair Journey team in 2012. This was when I first met them here in Porto in 2012. They were in an incredible building, and not in a positive sense. I think this is one of the kind of buildings that we would tear down in the Netherlands, but here they just kept it alive. They took 3 rooms, and they started to the Fair Journey story in 2012. So to be honest, when I came in, the environment didn't give me the best possible impression, with broken laptops and not even an ice machine to be seen anywhere. But the coffee machine was great, so I'm not complaining. But this was the team, and the team made a fabulous impression. The team was knowledgeable. The people were driven by trying to done, good antibody engineers, energy and enthusiasm all over the place. And if you've interacted with them in later stages, you probably know what I mean. And that made me, in 2012, decide to, I think, become the first customer of and start doing projects with them. And that is a collaboration that's still ongoing. So we just did some counting. So we have 9 years of collaboration. We ran 140 different studies, and we addressed 29 different targets together. And I'm using the word together because these guys know what they're doing, and they've been tremendously helpful in helping Mirus and collaborating with Mirus to achieve where we are as a company today. So we are proud of having been part of that journey, and some truly meant congratulations to the team. And we're looking forward to continue to work with you as we go along. So this is the end of my presentation. Thank you very much. Happy to take some questions.
Ton, thank you very much for wonderful talk. I must say that I was very impressed by your attempt to convince your investors that on a per data point basis, they haven't invested enough. That was very nice done. I have a serious question for you as well. So your system is high throughput, 10,000 of data points, but if you start combining all the variables, targets, orientations, linkers, affinities, you run out of 10,000 points very, very quickly. And you spoke about it in the context of biparatopic molecules, but in the context of a true bispecific, how densely do you need to sample to create a useful data with this 10,000 data points constraint?
Oh, you're absolutely right, and I think that problem even becomes worse with the triclonics approach, where you have the position of the binding arms. Is it on the short arm? Is it on the long arm? Is it on the outside or the inside? So you're absolutely right. We do make choices, right? We cannot be completely agnostic about all the variables and just combine them. It would take millions and millions of cultures to look at. But I'm hopeful that we will increase those numbers significantly. But still, you make choices. And choices are based on, what antibody engineers probably call intuition. You don't want everything to be with the same link and link. Let's create some flexibility. We learn quickly what works and what doesn't work. So if and that's also in biparatopics, to be honest. When certain arms work very well in these types of screens, they tend to work with many different ones. We've seen that in a HER2 or HER3 bispecific antibody, and they're seeing it again in these types of assays. So good arms tend to stay good arms even with other molecules associated in the second arm. So that makes you helpful that you, once in a while, you hit one of these good arms, you see many combinations that work, and you can keep working with that. But you're absolutely right. I mean, the numbers are staggering, so there's no way we can address all those combinations of variables.
Thank you, Tom. Yep. Questions from the audience? On the left, sixth row.
Do you hear me? Oh, thanks for your very nice presentation. So I have a question about your choice. You have decided to move in the direction of the tri-specific, which increased the complexity of the molecules. Potentially, it could be some disadvantage also with the immunogenicity increase. but you showed that you clearly improved the efficiency by targeting 2 TAAs. Have you compared if you only stay with the B-specific with 1 TAA, but making an effort to increase the affinity from nanomolar to picomolar, what happened, if it's comparable or not?
Yeah, I think the scary part of the solid tumor T-cell engager approach is, of course, that you're unleashing an incredibly potent mechanism on normal cells that may express lower levels, maybe, of the 2 targets. So that would create true havoc, because T-cells are true killers, and they can do a lot of damage on normal tissues, right? So the driver is not as much the efficacy, but rather the specificity and creating a true therapeutic window. So the avidity balance and the therapeutic window we're seeing in our models needs to translate into a dosing schedule and a final dose that makes sense in terms of turning T cells onto the tumors, on the solid tumor, and not onto the normal tissues. And that's to be seen, right? That's the holy grail for T-cell engagers for solid tumors. For the platform, actually, because I haven't talked much about CMC or immunogenicity, I mean, the bispecifics are like a regular monoclonal antibodies, the ease, the yields, the stability. There's nothing that differentiates them from a regular antibody. What we know about the triclonics now is that in terms of productivity and stability, well, they even seem to be a little better for reasons we don't understand. They haven't been in patients yet, so we don't know about PK and we don't know about immunogenicity. But the only thing in that molecule is a linker that's been used many times in other solutions for single-chain FEs or linker-based solutions. So we're not too worried about that. So again, the platform is a given. We think it's robust and very strong. But the challenge is here, indeed, can we actually selectively address tumor cells?
Thank you.
There was a question on the right, third row, and then there was a question in the 5th row after that.
Does this work? So the tri-specifics are quite interesting, and this probably also goes for Dragonfly. But so I used to work for Medarex, and Medarex, before it became a human antibody mouse company, was actually a bispecifics company, which what a lot of people probably don't know. And they went away from it because the pharmacodynamics were just impossible. And I think that's the question I have for cell engagers. I mean, how does this work in terms of getting the T cell, the tumor cell, and the antibody together in one place at the right time to do the right thing? I mean, there's been some papers suggesting you have to have pre-presence of T cells in the tumor for this to work at all. And I'm sort of wondering what your view is on how to tackle this and how to potentially screen for the best way to tune your affinities.
Yeah, I guess you're quite right that there are some unknowns about the solid tumors. And obviously, in a clinical trial strategy, you would focus on the tumors that have substantial T cell infiltrations. You're not trying to solve the issue of the immunological desert, as we call it. But you can also, as I briefly mentioned, for example, we've seen in multiple models that these tri-specifics and bispecifics actually can also attract new T cells into the tumor. We don't really know why that happens, but it's quite obvious. And of course, what you're trying to do in the tumor is create an inflammatory environment, because these T cells start to secrete cytokines that will start to trigger activation of cell types and expression of molecules that are relevant for amplifying the system. So I'm not going to see this as an easy approach, but as I mentioned, this is the future. This is where we need to go if we don't want to be stuck with T-cell engagers that we're only using for hematological tumors. And it is my conviction that that's what biotech needs to do, right? We need to just try to be successful in those areas and fail every once in a while. And as I said, still convince investors that we're on the right track. But yeah, you're quite right. We'll have to see. This needs to go into the clinic, and we'll see.
I mean, one follow-up question then on this one is, it looks like you would then have to have a hot tumor for this to work well. And you then have to think from a therapy perspective, how do I either get a tumor hot or use hot tumors to target? And
Sure.
that should factor into the strategy, I think, in developing the therapeutic approach rather than the drug itself.
No, I think we're seeing the early evidence of that is indeed going to happen. So I'm just referring to, for example, Regeneron, who's now putting 2 bispecifics together in one patient to separate priming of T cells and co-stimulation in a bispecific manner. But the future will be about these kinds of T cell engagers if they work in the, let's say, easy hot solid tumors, in approaches where we're trying to break that barrier with completely different approaches that really focus on other cells in the microenvironment that build these impenetrable walls for T cells. So combination strategies, if you're fine with this particular approach, you can envisage other approaches, bispecific or trispecific, where a second antibody will try to break down the barrier. And that's, of course, again, the holy grail, right? We try to do something with these immunologically cold tumors rather than sit and say, well, we can't do anything.
Thank you.
Let's thank Ton again for a wonderful presentation.